This little video is about transport in plants, but it's about the transport of sugars in the phloem rather than the transport of water in the xylem, which is what my last video was about. So I've got a little diagram here which I'll fill in as we go. But I've got, for context, a little diagram of a plant up here. And this bit here, it shows you, you'll be familiar from the last video that this is the xylem, but there's also phloem that runs alongside the xylem. And this bit here is represented by this section up here. Okay, so we have a leaf cell here, which is producing sugar through photosynthesis. And because it's producing the sugar, we say that it's a source cell. We also have this little cell here, which is called a companion cell. We'll come back to what they do later. And this is the phloem that's running alongside the xylem here. So they are a series of vessels, tubeways if you like, that run throughout the plant. The phloem vessels are sometimes called sieve tubes. So I might refer to them as sieve tubes throughout the tutorial. Okay, and then at the other end of the plant, we have this section here. So we have the cells that need the sugar that the leaf cell is producing. They need the sugar because they're going to use it for respiration. And so we refer to these cells, the ones that use the sugar, as sink cells. So what's happening is that in the plant, the leaf cells are making the sugars through photosynthesis, and these sugars have to be transported to other parts of the plant. And it's the phloem that transports the sugars dissolved in water. So the first thing that has to happen is that the sugars in the form of sucrose get transported into the companion cell. Okay, so this process has got a few stages. So if we just go down to the bottom of the page here, I have enlarged the leaf cell and the companion cell next to it. Yeah, and we're just going to draw a little diagram to show how the sucrose actually gets from the leaf cell into the companion cell. How do we get the sucrose out of the leaf cell into the companion cell? Pretty much it's active transport, but with a twist. Okay, so first of all, what has to happen is using ATP. So this would be active transport. The companion cell has to transport lots and lots of hydrogen ions into the leaf cell. And you might be thinking, why are we now talking about hydrogen ions when we should be talking about sucrose? Well, what we now have, it will connect up in a minute, what we now have is we have a very high concentration of hydrogen ions in the leaf cell. And so the hydrogen ions will tend to want to now diffuse back into the companion cell. Okay, so we can see the active transport, let's just label that, using the ATP, has transported the hydrogen ions into the leaf cell so that now the hydrogen ions can diffuse back into the companion cell. Well, we haven't mentioned the sucrose. What's that got to do with getting the sucrose out of the leaf cell into the companion cell? Well, what happens is the hydrogen ions, when they diffuse back into the companion cell down a concentration gradient, they take with them the sucrose. So what we have is we have an example of co-transport. So this is important because actually there's less sucrose here than there is here. And so this system allows the cells to get the sucrose from the leaf cell into the companion cell against its concentration gradient. Okay, So that's step one. How does the sucrose get out of the leaf cell into the companion cell? This is how it happens. Okay, let's go back up to our diagram here. Now what we're going to think about is once the sucrose has gone into the companion cell, which we've just talked about, it then enters the sieve tube, the phloem, through this little gap here. Now let's just give this a name. This little gap is called a plasmodesmata. Okay, so the sucrose is now going to enter the phloem. And as it enters the phloem, because it's soluble, it lowers the water potential inside the sieve tube, inside the phloem. So remember that the xylem is carrying water up towards the leaf cells. And it has a relatively higher water potential than the neighbouring sieve tube, the neighbouring phloem. And so when the sucrose enters and lowers the water potential, water will enter the sieve tube from the xylem by osmosis. Okay, so let's just recap that again. The sucrose has entered the phloem. The sucrose lowers the water potential, so it's now lower than the water potential inside the xylem. That means that water will enter the sieve tube from the xylem by osmosis. So we're getting an increased volume of water here. Let's just write that down because that's really important. So an increased volume of water in the phloem at this point, at the top of the plant, increases the pressure. Okay, so more water means there's going to be more water molecules pressing up against the walls of the sieve tubes, the walls of the phloem, and so that's going to increase the pressure of water at the top of the plant, at the top of the phloem. Meanwhile, let's look at what's happening at the bottom of the plant. The sucrose is being taken up into the companion cell and then entering the root cell. Okay, so the sucrose enters the cell and it lowers the water potential. Bit of a theme developing here. So what that means is the water follows from the companion cell and from the phloem, the water follows by osmosis. So now we've got water leaving the phloem. So we now have a lower volume. We have a lower volume of water at the bottom of the phloem. And of course, that's going to decrease the pressure. Okay, so let's recap. The sucrose enters the phloem, it lowers the water potential. That means that water comes into the phloem from the xylem down a water potential gradient. That means you've got more a higher volume of water, and so you're going to increase the pressure. Meanwhile, the sucrose enters the cell at the bottom of the plant, lowering the water potential, so water follows by osmosis. Water is now leaving the phloem at the bottom of the phloem. And so because of that, you get a lower volume of water and that decreases the pressure. We now have from the top of the phloem to the bottom of the phloem, from the top to the bottom, we have a pressure, not a water potential gradient, a pressure gradient. From top to bottom of phloem. Okay, so water enters the phloem here, water leaves the phloem here. Water entering increases the pressure, water leaving decreases the pressure. And so we have a pressure gradient higher here, lower here, and so the water gets pushed down along a pressure gradient. Down a pressure gradient. And of course, as it flows down, it takes with it anything that's dissolved in it, including the sucrose. So this is the way that the sucrose can be translocated from the leaf to the places where it's needed in the plants. In this case, it's going down to the root cells. And we call this the mass flow theory, because it's thinking about all of the water flowing down a pressure gradient and taking with it anything that's dissolved, like the sugars, translocating them from the top of the phloem to the bottom of the phloem.